Module 4 begins transcription and translation. This is cohesive with chapters 14 and 15 in your textbook. In general, we'll look at the two processes, gene transcription and protein translation. We'll look at how these processes differ in both pro and eukaryotic organisms, and start to really look at gene structure. So if we look at a prokaryotic cell, or bacteria, which has no membrane-bound organelles, versus a eukaryotic or a mammalian cell, where the nucleus is enveloped and all of our chromosomal DNA is located within the nucleus, you can start to see how these processes of transcription and translation differ. In prokaryotic cells, the process of transcription and translation, or the production of RNA and the production of protein, is coupled. These processes are not segregated at all within the cell. This differs significantly in mammalian cells, where the process of transcription happens within the nucleus, and a messenger RNA is mature and exported out of the nucleus, where it's translated in the cytosol of the cell by ribosomes. The separation of these two processes, transcription and translation, in eukaryotic cells is a key difference between eukaryotic cells and prokaryotic cells. So let's look at the schematic for a minute. We've looked at DNA, shown here in this schematic. This is our double-stranded DNA, where our top strand is our coding strand, or our sense strand, and the bottom strand, the 3' to 5' strand, is considered the template strand. This is because in our messenger RNA, we want it to be identical to our coding strand. You can see that here, where the coding strand has a sequence of ATG, which is seen here in messenger RNA as an AUG. Remember, that's because uracil replaces thiamine in the case of RNA. The use of the antisense strand as the template strand for transcription is integral to translating a protein product that is then consistent with our coding DNA strand. Again, the DNA template strand for transcription is the antisense strand of DNA, so that we can generate a messenger RNA strand, or mRNA strand, that carries the same sequence as our DNA coding strand. In all cases, thiamine in DNA is replaced by uracil in RNA. What you can also see here is that in the protein, every 3 bases, or what's referred to as a codon, is complementary to a single amino acid. This is a universal code where 3 nucleotides are specific for a single amino acid. So what are the roles of RNA within a cell? RNA is multifaceted. First, RNA functions as messenger RNA transcripts, and these encode proteins, like we saw on the previous slide. RNA makes up 2/3 of the translational machinery, meaning it's part of the ribosome, which is a protein RNA complex that facilitates protein translation. Likewise, protein translation requires tRNAs, or transfer RNAs. These bring amino acids to the ribosome during the translational process. RNA can also participate in the process of transcription to impact transport and splicing. This is referred to as a ribonucleotide protein, in which RNA is part of a protein and can participate in these processes. And RNA can also be used as hereditary material. This can be the case in viruses, and it can also be the case in transposons, like we saw previously. So if we look at the process of transcription, it uses a DNA strand as a template, and remember this is the antisense strand, and grows an RNA strand in the 5' to 3' direction using phosphodiester linkages similar to DNA replication. So how is this RNA strand synthesized? Unlike DNA replication, which just required an origin of replication, transcription is regulated by regulatory elements within each gene. Once transcription is initiated, the DNA unwinds and the RNA is transcribed in the 5' to 3' direction from the template. It's similar to DNA synthesis, except for the use of nucleotides rather than deoxynucleotides. There's no primer needed to initiate transcription, and you add uracil instead of thiamine. In this case, uracil complementary base pairs with adenine. And again, you use an RNA polymerase rather than a DNA polymerase. So let's highlight differences between pro and eukaryotic organisms. Prokaryotic organisms have a single RNA polymerase. This RNA polymerase transcribes all of the messenger RNA, transfer RNA, and ribosomal RNAs within the cell. In contrast, eukaryotes possess 3 RNA polymerases that have unique functions. RNA polymerase 1 transcribes ribosomal RNA, RNA polymerase 2 transcribes messenger RNAs and some small nuclear RNAs, whereas polymerase 3 transcribes primarily tRNAs. In general, there are 3 steps to transcription, and we'll focus on initiation and elongation. These processes occur in both pro and eukaryotes, and the elongation process is conserved between the two of them. Termination differs marginally between the two of them, but this will not be the focus for us. Let's take a look at eukaryotic and prokaryotic promoters to help identify how RNA polymerase aligns itself to start transcription. If this is denoted as a sequence of DNA, we can see that the transcriptional start site is here at +1. Elements that are said to be upstream of the start site are in this direction and are denoted in negative numbers. These numbers are relative to how far they are away from the transcriptional start site. Elements after the transcriptional start site are said to be downstream. In prokaryotic organisms, RNA polymerase and the sigma factors recognize 2 consensus sequences. The first consensus sequence is referred to as the Tata box, and this is the thiamine-adenine rich region, approximately 10 base pairs upstream from the transcriptional start. The other consensus sequence is the -35 sequence, which, as it implies, is -35 base pairs approximately upstream of the transcriptional start. Specifically, the -10 site is recognized by the sigma factor, which is required for recruitment of RNA polymerase and will help align RNA polymerase to the transcriptional start site. These are the basal consensus sequences required for the initiation of transcription. Additionally, there may be repressor binding sites and activator binding sites that will help stimulate or repress transcription. In the eukaryotic promoter, shown here, you can see that in general it reserves the same or similar consensus sequences. Upstream of the transcriptional initiation site, shown here, you have a conserved Tata box region and a region here denoted the BRE, which is a binding site for a transcription factor TF2 beta. This is a required transcription factor that is necessary for the action of RNA polymerase II. In eukaryotic organisms, transcription is regulated at several levels, and recruitment of a basal number of transcription factors and RNA polymerase are required for basal level of transcription. Additionally, there are proximal elements, shown here, as well as downstream elements, shown here, which can also increase or decrease the level of transcription. These are bound by what's referred to as gene-specific transcription factors. We'll talk about these more when we talk about regulation of gene transcription. Let's look more closely at the initiation process in prokaryotes. Again, this requires an RNA polymerase, which binds with a sigma factor, to create an RNA polymerase holoenzyme. The sigma factor specifically recognizes the Tata box, and this initiates the transcriptional process. Sigma factor is required for efficient binding and transcription, and depending on the consensus sequence of the Tata box, there are different sigma factors that can recognize these different promoter sequences. Once bound, the RNA polymerase can act as its own helicase and unwinds the DNA, binding tightly to the -10 site and more loosely to the -35 site. And again, there are different types of sigma factors, and these can influence the level and dynamic of gene expression, meaning they can regulate, to a certain degree, the level of transcription. After a few base pairs of RNA has been synthesized, the sigma factor is released and recycled for other reactions. At this point, the RNA polymerase can complete the transcription at a rate of 30 to 50 base pairs per second. As it unwinds the DNA rapidly reanneals behind the enzyme after transcription, and most of the RNA is displaced as this double helix forms. What you can see here is this is our promoter region, which includes our consensus binding sequence sites, and downstream of that are several structural genes in tandem. The transcript generated is considered a polycistronic mRNA, meaning it encodes for several independent proteins. This is shown here, where 3 proteins are encoded by this polycistronic mRNA. This is not the case in eukaryotes, which generate a monocistronic mRNA. Let's look at this question. For the process of transcription, the template strand is the sense strand of DNA, antisense strand, or either strand of DNA. Let's revisit this figure. In this case, the template strand is always the antisense strand. The correct answer here is B. The antisense strand is required such that the mRNA is an exact copy of the coding DNA strand, except for the replacement of uracil in the RNA transcript.